Welcome to a deep dive into the fascinating and often overlooked world of microbial biochemistry.
Yeah.
You sent over some really interesting sources on this topic, and I have to say, even I'm already learning a ton just skimming through them.
Oh, wow.
Get ready for some serious aha moments as we unravel the secrets of these tiny but mighty organisms.
It is amazing how much complexity is packed into these microscopic wonders.
It is.
I'm excited to kind of break down these complex concepts and really explore how microbes are built, what they're made of, and how we can even use biochemistry to identify them.
It's like detective work, but on a molecular scale.
Absolutely.
Speaking of molecules, let's start with the fundamentals.
Okay.
The building blocks of life.
Right.
I'm seeing a lot about carbon in these sources.
Yeah.
What makes it so special?
Carbon is truly the backbone of life. Think of it as the ultimate tinker-toy connector.
Oh, yeah.
Each carbon atom can form bonds with up to 4 others.
Wow.
This allows carbon to assemble into a mind-blowing array of structures. Straight chains, branched chains, rings, you name it, carbon can do it. It's incredibly versatile.
So carbon provides the framework, the scaffolding upon which the incredible diversity of life is built.
Yeah.
But how do we get from simple carbon structures to the complex molecules that make up living organisms?
Well, just like with tinker toys, you can use the same basic building blocks to create endless variations.
Yeah.
That's where isomers come in.
Oh, isomers.
They're molecules with the same atoms, but arranged differently in space, leading to drastically different properties. It's like rearranging furniture in a room.
Uh-huh.
Same pieces, different look and feel, and definitely different functionality.
So even a slight change in arrangement can have a huge impact on the molecule's behavior.
Again.
It makes you realize that structure dictates function at every level of biology.
Absolutely.
Now, these sources also mention functional groups.
Yeah.
What role do they play in this molecular construction set?
Functional groups are like adding specialized attachments to our molecular tinker toys. They're specific clusters of atoms that attach to the carbon framework and give molecules their unique characteristics and reactivity. It's like each functional group has its own superpower, influencing how the molecule interacts with others.
Okay, I'm starting to see how this all comes together.
Right.
Carbon provides the foundation, isomers create diversity, and functional groups add specialized functions.
Yeah.
It's a brilliant system.
It is.
But how do we go from these individual molecules to the larger macromolecules that make up cells?
That's where dehydration synthesis comes in.
Okay.
It's a fascinating process where smaller molecules called monomers link together to form long chains called polymers. During this process, a water molecule is released. It's like 2 molecules joining hands and letting go of a water balloon as they do.
Oh, wow.
This process of linking monomers is essential for building the macromolecules that are crucial for life. Carbohydrates, lipids, proteins, and nucleic acids.
So it's like building a chain link by link with each link representing a monomer.
Yeah.
Now let's zoom in on carbohydrates. They often get a bad rap.
They do.
But there's a lot more to them than just sugar, right?
Absolutely.
Okay.
Carbohydrates are the most abundant biomolecules on Earth, and they play essential roles in energy storage structure and even cellular communication. Think of them as the fuel-building materials and messengers of the microscopic world.
Okay, so carbohydrates are much more versatile than I initially thought.
Yeah.
Now I'm seeing terms like monosaccharides, disaccharides, and polysaccharides.
Okay.
What's the connection between all of these?
It's all about hierarchy.
Oh, right.
Monosaccharides are the simplest sugars, the single units like glucose or fructose.
Got it.
Think of them as the individual beads on a necklace. Disaccharides are formed when 2 monosaccharides link together, like sucrose, which is table sugar.
Yeah.
Imagine 2 beads joined together.
Okay.
And finally, polysaccharides are the long chains composed of many monosaccharides linked together.
Right.
Think of it as a whole strand of beads, sometimes hundreds or even thousands of units long.
It's like building a sugar chain with each type of carbohydrate representing a different level of complexity.
Exactly.
But some polysaccharides store energy while others are structural, right?
Yeah, yes.
What's the difference?
You got it.
Okay.
Starch found in plants like the potatoes you had for dinner and glycogen stored in our muscles and liver are energy storage polysaccharides. They're like the body's pantry, storing glucose for later use. On the other hand, cellulose, which gives plants their rigidity, and chitin, which forms the exoskeletons of insects and the cell walls of fungi, are structural polysaccharides. They're like the scaffolding that holds everything together.
So the same basic building blocks, sugars can create everything from a potato to a beetle shell.
They can.
It really highlights the elegance and efficiency of nature's design.
Absolutely.
Now let's move on to another essential macromolecule that often gets a bad rap.
Okay.
Lipids.
Lipids.
Are all lipids bad?
Definitely not.
Okay.
Lipids are incredibly diverse and play essential roles in energy storage, insulation protection, and even cell signaling. They're like the body's multi-talented performers with a role for every occasion.
So it's more nuanced than just fat equals bad.
It is.
But what exactly are lipids made of? They seem so different from carbohydrates.
While lipids are mostly made of carbon and hydrogen, like carbohydrates, they have a distinct characteristic.
Okay.
They're generally hydrophobic, meaning they don't mix well with water. Think of oil and water.
Right.
They just don't want to mingle.
Yeah.
This hydrophobic nature is key to their function, especially when it comes to forming cell membranes.
That makes sense.
Yeah.
Cell membranes need to act as barriers separating the watery environment inside the cell from the watery environment outside.
Right.
But how do lipids create this barrier? I'm seeing a lot about fatty acids in these sources.
Okay.
How do they fit into the picture?
Fatty acids are the building blocks of many important lipids. They consist of long chains of hydrocarbons, which are essentially chains of carbon atoms studded with hydrogen atoms.
Okay.
These hydrocarbon chains are what make lipids hydrophobic.
Okay.
Now, fatty acids can be saturated or unsaturated depending on the types of bonds between carbon atoms.
Right, like the difference between butter, which is solid at room temperature and rich in saturated fats.
Yes.
And olive oil, which is liquid at room temperature and rich in unsaturated fats.
Exactly.
But how do these fatty acids contribute to the formation of cell membranes?
Well, fatty acids team up with glycerol, a small alcohol molecule, to form some of the most essential lipids, like triglycerides and phospholipids. Triglycerides are the superstars of energy storage.
Okay.
Think of them as the body's long-term energy reserves.
Got it.
They pack a lot of energy in a compact form thanks to those long hydrocarbon chains.
So triglycerides are like the body's high-octane fuel depot.
Yeah.
What about phospholipids?
Right.
How do they contribute to building those all-important cell membranes?
Phospholipids are truly fascinating molecules. Yeah.
They're like the architects of cell membranes, thanks to their unique amphipathic nature.
Okay.
This means they have both hydrophobic and hydrophilic regions, parts that fear water and parts that love water.
Huh.
Imagine a molecule with two long water-fearing tails and a water-loving head.
So it's like a tiny molecular Janus with two faces looking in opposite directions.
I got it.
How does this dual nature allow phospholipids to form membranes?
It's all about how they arrange themselves.
Right.
Picture a double layer of phospholipids with their hydrophobic tails tucked together, shielded from the surrounding water, and their hydrophilic heads facing outward, interacting with the water inside and outside the cell.
Okay.
This arrangement creates a stable barrier that defines the cell's boundaries and controls what goes in and out.
That's a brilliant design.
It is.
It's amazing how such a simple concept, having both water-loving and water-fearing parts, can lead to such a complex and essential structure.
Right.
But the world of lipids doesn't stop there, does it?
No, it doesn't. I'm also seeing mentions of isoprenoids, waxes, and steroids. What are their roles?
Isoprenoids are a diverse group of lipids that play roles in everything from pigmentation, think of the vibrant colors of flowers, to hormones.
Oh, wow.
Think of cholesterol, which is essential for cell membrane function.
Right.
Waxes are long-chain lipids that are typically water-resistant, providing protection and waterproofing.
Okay.
Think of the waxy coating on fruits that helps prevent water loss.
Okay, so waxes are like nature's raincoats.
Exactly.
And what about steroids? I know cholesterol is a steroid and it often gets a bad rap.
It does.
But aren't steroids important for our bodies too?
Absolutely.
Okay.
Steroids are a unique class of lipids with complex ring structures. Cholesterol is a key component of animal cell membranes, helping to maintain their fluidity and integrity.
Right.
But it's not just animals that rely on steroids.
Oh.
Bacteria and fungi have their own versions of cholesterol, like hopanoids in bacteria and ergosterol in fungi, which also play structural roles in their membranes.
So even though these microorganisms are very different from us, they share some fundamental building blocks.
We do.
It's like we're all part of the same molecular family.
Right.
Now, we've talked about carbohydrates and lipids, but what about proteins?
Yes.
They seem to be involved in almost everything.
Proteins are truly the workhorses of the cell. They catalyze reactions, transport molecules, provide structural support, and play countless other roles.
Wow.
They're essentially chains of amino acids linked together.
Okay.
Imagine a string of beads with each bead representing a different amino acid.
Amino acids.
Yeah.
I remember those from biology class.
Yeah.
They're the building blocks of proteins, right?
They are.
But what makes them so special?
Wow.
How do they contribute to the incredible diversity of protein function?
Each amino acid has a central carbon atom bonded to 4 different groups.
Okay.
A hydrogen atom, an amino group, a carboxyl group, and a unique side chain called the R group. It's this R group that gives each amino acid its individual personality, influencing its properties and how it interacts with other molecules.
So it's like each amino acid has its own unique set of skills and talents.
Right.
Contributing to the overall function of the protein.
That's a great way to put it.
Okay.
And when these amino acids link up via peptide bonds, they form peptides, which are short chains of amino acids. As more and more amino acids join the party, we eventually get the mighty proteins.
So it's like building a complex machine from smaller, specialized parts.
It is.
But proteins aren't just long, floppy chains, are they?
No, they're not.
They fold into intricate, three-dimensional shapes.
You're absolutely right.
Okay.
The sequence of amino acids in a protein, called its primary structure, determines how it folds into these intricate 3D shapes.
Got it.
These folds are crucial for protein function, like creating the active site of an enzyme or the binding site of a receptor.
I've heard about different levels of protein structure.
Yes.
What are those all about?
Think of it like origami. With each fold adding complexity and functionality, the primary structure is just the linear sequence of amino acids.
Right.
Then we have secondary structure, where local interactions between amino acids cause the polypeptide chain to twist and fold into alpha helices, which look like spirals, or beta sheets, which resemble pleated sheets.
So it's like the polypeptide chain is getting organized into these repeating patterns.
It is.
What happens next?
Then we move on to tertiary structure, which is the overall 3D shape of a single polypeptide chain, formed by interactions between different parts of the chain. This is where things get really complex, with loops, bends, and folds all contributing to the protein's unique shape.
Wow.
Finally, some proteins have quaternary structure, which arises when multiple polypeptide chains come together to form a functional protein complex. Imagine several origami creations coming together to form a larger, even more intricate structure.
Wow. So it's like building a super protein from individual protein subunits.
Exactly.
But this intricate folding must be delicate. What happens if a protein's environment changes? Like if it gets too hot or too acidic?
You're right. Protein structure is sensitive to its environment. If a protein encounters extreme temperatures or pH changes, it can denature.
Okay.
This means it loses its higher level structure and, as a result, its function.
Got it.
Think of a cooked egg white.
Right.
The heat denatures the proteins, causing them to unfold and solidify.
That's a great example. It's like the protein is losing its carefully crafted shape, and with it, its ability to do its job.
Exactly.
Now, before we move on, I've also come across the term conjugated proteins. What sets them apart?
Conjugated proteins are fascinating because they have non-protein components attached to them.
Okay.
For example, glycoproteins have carbohydrates attached, while lipoproteins have lipids attached. These additional components often play important roles in the protein's function, especially in cell membranes, where they can act as receptors or signaling molecules.
So it's like adding special attachments to the protein to give it even more abilities.
Yeah. It really highlights the remarkable complexity and modularity of biological systems.
Exactly.
Yeah.
Conjugated proteins showcase the incredible diversity and complexity of the molecular world.
Wow.
And this complexity is amplified when we consider the vast array of proteins found in a single cell, each with its own unique structure and function, all working together to orchestrate the symphony of life.
It's incredible.
It is.
It's mind-boggling to think about the intricate dance of molecules happening inside every single cell.
It is.
But all this talk about the building blocks of life has me thinking about how scientists actually identify these microscopic suspects. I'm intrigued by this idea of microbial forensics.
Yeah.
It's like CSI,
It is.
but at the microbial level.
At the microbial level.
So instead of dusting for fingerprints, we're analyzing molecules to solve microbial mysteries.
Exactly.
What are some of the techniques scientists use to unveil the identity of these microscopic culprits? Well, one approach is phenotypic identification, which relies on observing the microbe's physical and biochemical traits.
Okay.
Scientists can analyze the types of lipids, sugars, and storage compounds a microbe produces, giving them clues about its identity.
So it's like creating a microbial profile based on its lifestyle and preferences.
It is.
What kind of information can these biochemical clues reveal?
Well, for example, analyzing the types of fatty acids present in a microbe's cell membrane can help determine its species.
Oh.
Some microbes prefer to feast on certain sugars, while others thrive in specific environments. All these preferences leave telltale signs in their biochemical makeup.
It's fascinating how a microbe's lifestyle choices are reflected in its molecular fingerprint.
It is.
But phenotypic identification seems like it might be a bit time-consuming.
Yeah, it can be.
Are there faster, more high-tech methods available?
Absolutely.
Okay.
One technique that's revolutionized microbial identification is MALDI-TOF, which stands for Matrix-Assisted Laser Desorption Ionization Time of Flight Mass Spectrometry.
Oh my goodness.
It sounds complicated,
It does.
but the basic idea is quite elegant.
Okay, break it down for me. How does this MALDI-TOF technique work?
So imagine you have a sample of microbes.
Okay.
Scientists mix the sample with a special matrix and then zap it with a laser. This vaporizes the molecules in the sample, creating ions that are then separated based on their mass-to-charge ratio.
Okay.
The result is a unique spectrum, like a molecular fingerprint, that can be compared to a database of known microbial spectra to identify the species.
So it's like creating a unique barcode for each microbe based on the weight of its molecules.
Yeah, it is.
That's incredible.
It's an amazing technique.
And it's incredibly fast and accurate, allowing scientists to identify microbes in a matter of minutes.
Yes.
This is invaluable for diagnosing infections, tracking outbreaks, and monitoring environmental samples.
Absolutely.
It's amazing how technology is allowing us to delve deeper and faster into the microbial world.
It is.
Are there other techniques that use this kind of molecular detective work?
There are.
What do they-
Another powerful method is FAME analysis, which stands for fatty acid methyl ester analysis.
Okay.
This technique focuses on identifying microbes based on their unique fatty acid profiles.
So you're telling me that the types of fats a microbe produces can reveal its identity.
I am.
That's remarkable.
It is.
How does that work?
Well, each microbial species has a distinct composition of fatty acids in its cell membrane.
Okay.
Scientists can extract these fatty acids, convert them into methyl esters, and then analyze them using gas chromatography. This creates a chromatogram, a kind of molecular fingerprint that can be compared to a database to identify the microbial culprit.
It's like analyzing the microbial equivalent of a dietary fingerprint.
Exactly.
Are there any other techniques that take a similar approach?
Yes. PLFA analysis, which stands for phospholipid fatty acid analysis, is another valuable tool that focuses specifically on the fatty acids found in phospholipids, the key building blocks of cell membranes. This technique is particularly useful for studying microbial communities in environmental samples such as soil or water.
It's incredible how much information is hidden within these tiny molecules.
It is.
It's like each microbe carries a secret code that reveals its identity and lifestyle.
Right.
Now, I've also come across the term proteomic analysis.
Okay.
How does that fit into the world of microbial forensics?
Proteomic analysis takes a broader approach.
Okay.
It involves studying all the proteins present in an organism, its proteome, to pinpoint its identity and understand its metabolic capabilities.
So it's like taking a comprehensive inventory of all the protein workers in the microbial factory.
Yeah, you could say that.
By analyzing the proteins present, scientists can gain insights into the microbe's functions, its evolutionary relationships, and even its potential to cause disease.
Exactly.
This is especially powerful when combined with genomic analysis, which studies the microbe's DNA sequence.
Absolutely.
It's like piecing together a complete picture of the microbe's identity and capabilities from its genes to its proteins.
It is.
Are there any other techniques that stand out in this field of microbial forensics?
Yeah, another fascinating technique is Lancefield grouping, which is often used to classify different species of Streptococci.
Okay, so how does Lancefield grouping help differentiate between these different troublemakers?
Well, this method uses antibodies, which are like tiny molecular detectives, to identify specific proteins on the surface of Streptococci. These surface proteins act like name tags, allowing scientists to classify Streptococci into different groups based on their antibody binding patterns.
So it's like using antibodies to sort these bacterial suspects into different lineups based on their molecular closing.
Yeah, that's a good analogy.
This is all so incredible.
It is a fascinating field.
It seems like scientists have a whole arsenal of biochemical tools at their disposal to unveil the secrets of the microbial world.
It's true, microbial forensics is a rapidly evolving field, constantly developing new and more sophisticated methods to identify and characterize microbes. This knowledge is crucial for understanding the role of microbes in health disease and the environment.
It's amazing how these tiny organisms invisible to the naked eye can have such a profound impact on our lives and the world around us.
It is amazing.
This deep dive has definitely given me a newfound appreciation for the power of microbes and the scientists who are working to unravel their secrets.
I agree. It's a field that's constantly evolving with new discoveries being made all the time. What's particularly exciting is how this knowledge is being applied to solve real-world problems.
That's what I find so fascinating about science.
Yeah.
It's not just about understanding the world, but also about using that understanding to make a difference.
Right.
What are some of the areas where microbial biochemistry is having the biggest impact?
Well, one area that's particularly promising is medicine.
Okay.
Understanding the biochemical pathways of microbes is crucial for developing new antibiotics, antiviral drugs, and vaccines.
That makes sense. If we can understand how microbes function at a molecular level, we can find ways to disrupt those processes and stop them from causing disease.
Precisely, and it's not just about fighting disease. Microbial biochemistry is also playing a key role in developing new diagnostic tools.
Oh, wow.
For example, researchers are developing rapid tests that can detect the presence of specific microbes based on their unique biochemical signatures.
So instead of waiting days for lab results, we could potentially diagnose infections within minutes.
Exactly.
That would be a game changer.
It would, and the applications extend far beyond medicine.
Oh, wow.
Microbial biochemistry is also revolutionizing agriculture.
How so?
For example, scientists are studying the microbes that live in the soil, known as the soil microbiome, to develop more sustainable farming practices.
So we can harness the power of microbes to help plants grow better and healthier.
Precisely, some microbes can fix nitrogen from the air, making it available to plants, while others can help plants absorb nutrients more efficiently. Understanding these processes can help us reduce the need for synthetic fertilizers, which can have harmful environmental effects.
It's amazing how microbes can be our allies in creating a more sustainable future.
It is.
What are some other areas where microbial biochemistry is making a difference?
Another exciting area is bioremediation, which uses microbes to clean up environmental pollutants.
Okay.
Some microbes have the remarkable ability to break down toxic substances such as oil spills or industrial waste into harmless compounds.
It's like having a microscopic cleanup crew that can tackle even the toughest environmental challenges.
Exactly, and as we continue to explore the diversity of the microbial world, we're likely to discover even more applications for their biochemical superpowers.
This deep dive has definitely left me wanting to learn more about the hidden world of microbes.
Yeah.
What's sticking with you after exploring all this fascinating information?
Well, what always strikes me is the sheer ingenuity and efficiency of microbial life. These tiny organisms have evolved over billions of years to thrive in every imaginable environment, from the depths of the ocean to the human gut.
Yeah.
They've developed intricate biochemical pathways to extract energy, build complex structures, and interact with their surroundings. It's a testament to the power of evolution and the endless possibilities of life.
I couldn't agree more. This deep dive has been a journey into a world we often take for granted.
Right.
A world teeming with unseen life that plays a vital role in shaping our planet and our own existence.
It's a world full of wonder, mystery, and endless possibilities for discovery.
So after this deep dive into the world of microbial biochemistry, what's sticking with you? What surprised you the most? What new questions are brewing in your mind? Keep those questions simmering, because exploring the world of microbes is an adventure that never ends.